Welcome, medical microbiologists, to the Deep Dive. Today we're plunging into the complex world of an organism that holds a truly unique, almost contradictory position in the microbial world. It's celebrated as an environmental hero, yet, unexpectedly, it can be a silent threat right here in the clinic. We're taking a deep dive into Bacillus thuringiensis, or Blighty as you probably know it. This microbe is a powerhouse biopesticide, recognized globally for its effectiveness. But for us in the lab and clinic, it's also an opportunistic human pathogen, capable of causing severe, life-threatening infections, albeit rarely. Our mission today is to cut through the noise and deliver the essential knowledge you need to understand, identify, and manage Blighty infections. We'll be extracting the most important nuggets from a comprehensive review compiled using information sourced via Gemini Deep Search, with images provided by Microbicanvas. And remember, for a detailed list of primary sources and references, you can always consult the accompanying notebook document.
It's a truly fascinating organism. Its story really highlights the nuances of microbial identification and the critical role of the clinical microbiologist in guiding therapy, especially, when traditional guidelines are scarce. So yeah, let's unpack this complex microbe.
Okay, let's start broad. To truly appreciate Bacillus thuringiensis, we need to get the big picture. What makes this organism such a unique puzzle for us, and what are the initial challenges it throws our way?
Well, at its core, Bestie is a large, gram-positive, spore-forming bacillus. Pretty standard stuff there. But its unique position really comes from this dual identity. On one hand, it's globally celebrated as an effective, environmentally benign biological pesticide, drives a multi-billion-dollar industry. But concurrently, and this is where it gets interesting for us, it's an opportunistic human pathogen, capable of severe, life-threatening infections, though thankfully these are rare. The central challenge for medical microbiologists, I'd say, lies in its near-identical phenotypic and genotypic similarity to Bacillus cereus. It's really hard to tell them apart sometimes. And that key differentiator, the insecticidal crystal, the cry, and cytolytic, sit that proteins, they're plasmid-encoded, not chromosomal.
Okay, so that's not part of the core genome.
Exactly, and this makes clinical identification and risk assessment particularly tricky. It demands a deeper understanding beyond just standard lab practices.
So, despite its widespread environmental use as a pesticide, we absolutely need to take it seriously in specific clinical contexts. What are the main issues we'll be covering today that directly impact patient care?
Absolutely. While its insecticidal toxins are generally harmless to humans, BEAT can cause devastating infections in vulnerable individuals. That's point one. Point two, diagnosis and management are hampered by a striking lack of specific clinical guidelines and standardized lab methods, especially for AST, antimicrobial susceptibility testing. And crucially, its intrinsic resistance to all beta-lactam antibiotics. That's a non-negotiable therapeutic consideration we have to talk about. Finally, the resilience of its endospores. Standard infection control protocols often fall short, which is a critical point for our hospitals.
You've highlighted how challenging it is to distinguish Bacillus. That leads us right to its family tree, the Bacillus cereus group. Understanding this complex family is paramount for us in the lab, isn't it?
It certainly is. So Bacillus thuringiensis is a well-defined species within the bacterial domain. Phylum Bacillota, class Bacilli, order Bacillales, family Bacillacea, genus Bacillus. And as you said, it belongs to the larger Bacillus cereus group. It was first described way back in 1915 by Ernst Berliner. What's fascinating here is its membership in the Bacillus cereus sensilato group. This group includes 7 to 8 species that are so closely related they're often just considered a single complex. This includes not just Bacillus cereus, the common cause of foodborne illness, but also Bacillus anthracis, the agent of anthrax, so important neighbors.
Right, so differentiating them must be a real headache in the lab if they're so genetically and phenotypically similar. What are some of the common pitfalls?
Precisely. Their genetic and phenotypic overlap is extensive. They share a highly conserved chromosomal backbone. This makes differentiation using traditional biochemical tests or even 16S rRNA gene sequencing difficult. Unreliable, really. An isolate might be identified simply as B cereus group, or worse, misidentified as B cereus when it's actually B cereus.
And that could have real consequences.
Huge implications for both clinical management and public health epidemiology. That crucial exposure history to biopesticides might just get overlooked if the organism isn't correctly identified. The key differentiator for BAMT, remember, isn't chromosomal. It's the presence of those accessory genetic elements, specifically large plasmids carrying the cry and or site genes.
Ones encoding the endotoxin.
Exactly. If a strain loses these plasmids, it becomes functionally indistinguishable from B. cereus. So B is effectively a functional or maybe a pathovar designation. The human virulence factors, things like enterotoxins, are typically chromosomal and shared with B. cereus.
That distinction with the plasmids is really important. Can you maybe give us a quick comparison, just the key features that help us tell BN apart from B. cereus and B. anthracis?
Sure, yeah. Let's break it down quickly. Motility. B. anterior is motile, just like B. cereus. B. anthracis, though, is non-motile. That's a classic one.
Okay.
Hemolysis on sheep blood agar. Both B and B. cereus show beta hemolysis. B. anthracis is non-hemolytic, another good differentiator.
Yes.
Capsule production. Bt and B. cereus are non-capsulated. B. anthracis is the odd one out here. It's capsulated with poly-D-glutamic acid.
The anthrax capsule, right?
Yep. Key toxin plasmids. B has those chrysip plasmids for insecticidal action. B. cereus has enterotoxin and emetic toxin genes, chromosomal or plasmid. B. anthracis has PXO1 and PXO2 for its specific toxins and capsule. Penicillin susceptibility. This is critical. Both Bt and B. cereus are intrinsically resistant. B. anthracis is typically susceptible. And finally, perisporal crystal, present in B. antor, crucially absent in both B. cereus and B. anthracis.
You mentioned that perisporal crystal again. That sounds like the critical piece of the puzzle for identification in the lab. Is that truly the smoking gun we look for?
It absolutely can be, microscopically at least. It's a defining feature.
And what about different types or serovars of Bt? Does that matter clinically?
Well, BS is incredibly diverse. There are dozens of serovars based on flagellar or H antigens, like B. thuringiensis, subs B. israelensis for mosquitoes, Christaki for caterpillars. But for the clinical lab, serovar identification is really of limited value in assessing human infection risk. The human virulence factors are broadly conserved across the group, independent of the insecticidal profile. The crystal itself, though, tells you it's BS.
Okay, so with this challenging taxonomy, what does identification actually look like in our medical microbiology lab day to day? Let's start with microscopy and staining.
Right, microscopy is always the first step. Gram stain. You'll typically see large, robust gram-positive rods, about 1.0, 1.2 meters wide, 3.0, 5.0 meters long. Often in pairs or short chains, ends might look squared or rounded.
Pretty typical bacillus look then.
Exactly. Endospores. Like other bacillus, blue produces resilient endospores, usually cylindrical or ellipsoidal, central to paracentral. Importantly, they don't cause significant swelling of the sporangium. Green with spore stains like Schaefer-Fulton, perisporal crystals. And here's where it gets really interesting and clinically actionable. The pathognomonic feature, that protein perisporal crystal inclusion, visible adjacent to the endospore during sporulation.
Those are the delta endotoxins we talked about.
Precisely. They vary in shape, best seen with phase contrast microscopy or specific crystal stains. Honestly, seeing these crystals is the single most reliable microscopic feature to differentiate B from B. cereus. It's a real aha moment.
That's a great visual clue. How does it grow in culture? What do we see on our standard lab media?
Root grows readily on standard non-selective stuff. Growth conditions. It's a facultative anaerobe, grows best with oxygen. Mesophilic, optimal temps 25 degrees C to 37 degrees C. Standard incubator conditions are fine. Colony morphology. On sheep blood, agar, SBA, or tryptic soy agar, TSA, colonies are typically large, spreading, irregular, often have a dull, matte, or kind of frosted glass surface texture. Sometimes you even see new colonies growing on top of older ones.
Interesting. And hemolysis?
Hemolysis. Most strains are beta hemolytic on SBA. That's a key characteristic shared with B. cereus, helps differentiate from the non-hemolytic B. anthracis. Selective media. On MYP agar, mannitol egg yolk polymyxin, it produces pink to red colonies, no mannitol fermentation with a dense precipitation zone from lecithinase. On Pimba agar, colonies appear peacock blue with an egg yolk positive zone. But crucially, the colony morphology on these selective media, it looks identical to B. cereus.
So biochemical tests give us some clues. They confirm it's in the B. cereus group, but don't reliably separate B from B. cereus.
That's generally true. Key reactions. Catalase positive, oxidase usually negative, but some reports say positive, motility positive, beta hemolysis positive, lecithinase positive, mannitol fermentation negative.
Okay, standard B. cereus group stuff.
Right. Voges Proskauer positive, methyl red negative, citrate positive, starch hydrolysis positive, gelatin hydrolysis positive, casein hydrolysis positive, nitrate reduction positive, indole negative. It fits the profile.
But let's circle back to something you flagged as critical earlier. It's intrinsic resistance. Can you really elaborate on that? How profound is this for treatment?
This is where it gets really critical for clinical management, potentially life-saving, intrinsic antibiotic resistance. Like all members of the B cereus group, BS has a chromosomally encoded metallo-beta-lactamase called BC-Ni, and often other beta-lactamases too. This confers intrinsic resistance to all beta-lactam antibiotics, and I mean all of them. Penicillins, aminopenicillins, all generations of cephalosporins.
Even carbapenems.
Yes. Even if in vitro tests suggest carbapenem susceptibility, their clinical efficacy is highly unreliable because of that MBL. Any in vitro result suggesting any beta-lactam susceptibility should be viewed with extreme skepticism.
So labs should override it.
Absolutely. It should likely be overridden to resistant for clinical reporting. This inherent resistance is arguably the single most important piece of information the laboratory can provide for guiding empiric therapy.
Fair.
Full stop.
Wow. Okay. So given its environmental ubiquity and that built-in resistance, how does broad actually cause disease in humans? And who is most at risk?
Right, it's everywhere, but disease is rare. Barrett's pathogenicity in humans is almost entirely dependent on the host's underlying condition. Healthy people. Exposed to massive doses, usually no adverse effects. However, in immunocompromised patients, neonates, IV drug users, or those with indwelling medical devices, that's where it can cause severe disease. The main clinical syndromes we see with invasive Barrett infection include bacteremia and sepsis. Most severe, most frequently reported, almost exclusively in highly vulnerable patients. Think hematologic malignancies during neutropenia, neonates, central venous catheters. Onset can be fulminant, progressing rapidly to fatal septic shock.
Scary stuff.
Ocular infections. It's a recognized and particularly virulent cause of post-traumatic endophthalmitis, often from soil-contaminated objects hitting the eye or corneal ulcers. It can have a rapid destructive course, poor visual prognosis, endocarditis. Rare, but reported, primarily in IV drug users or patients with prosthetic heart valves, intracardiac devices. Often needs prolonged therapy, usually surgery too. Wound and soft tissue infections. From contaminated traumatic wounds, surgical sites, burns, can lead to cellulitis, abscesses, even necrotizing fasciitis in some cases. Other invasive infections, we've seen isolated cases, pneumonia in the immunocompromised, meningitis linked to ventricular shunts, even periodontitis.
That's quite a range. But when it comes to how it actually causes disease in humans, what's the mechanism? Are those famous insecticidal toxins involved at all?
That's a really important question. It's easy to assume the insecticidal toxins are the key players, but they're not considered major virulence factors in human infections. The primary CRA and CITIC toxins need specific conditions, alkaline pH, specific receptors in insect guts that just aren't met in humans.
So what is causing the damage then?
It's mediated by the same arsenal of virulence factors produced by B cereus. Things like membrane-damaging toxins, hemolysins, phospholipases, proteases, and critically, the ability to form robust biofilms, especially on medical devices. These biofilms shield the bacteria from immune responses and antibiotics, leading to persistent bacteremia, making infections incredibly tough to clear.
What about foodborne illness? I know you said B cereus causes it, but I feel like I've heard B mentioned too. Is that link real?
Ah, yes. Its role as a bona fide foodborne pathogen is indeed a subject of ongoing debate. It's controversial. Regarding emetic syndrome, the vomiting kind, the link is very, very tenuous. The syndrome is caused by a toxin called cerulide, which needs the CES gene cluster. That cluster is largely absent in most commercial beet strains used as pesticides.
So reports linking beet to vomiting might be?
They're often based on just finding bait alongside vomiting without definitive proof. It's much more likely due to B cereus co-infection or another agent entirely. However, for diarrheal syndrome, this association is more plausible. It's caused by heat labile enterotoxins, which are common throughout the entire B cereus group, including bait.
But can we tell them apart in practice?
Practically impossible without advanced molecular subtyping. So from a clinical standpoint, if it's diarrheal illness caused by the B cereus group, differentiating B from B cereus isn't usually feasible or clinically necessary. You treat the symptoms.
Okay, that makes sense. Now, given that fundamental intrinsic resistance to beta-lactams, how do we approach antimicrobial therapy for actual invasive bee infections?
Right treatment. It's guided by that predictable intrinsic resistance and, honestly, by clinical data extrapolated from the broader B cereus group. Formal evidence-based guidelines specifically for beet are largely lacking. So job one is remembering the intrinsic resistance to all beta-lactams, penicillins, cephalosporins. They're out.
What is typically susceptible then, based on testing?
Generally, in vitro testing shows susceptibility to glycopeptides like vancomycin, lincosamides like clindamycin, aminoglycosides like gentamicin, fluoroquinolones like Cipro and Levo, and tetracyclines. Linezolid and daptomycin have also been used successfully in reported cases, but experience is more limited.
So what are the international recommendations, or maybe the consensus based on B cereus group management for these serious infections?
Okay, for foodborne syndromes, just supportive care, rehydration, antibiotics aren't indicated. But for severe invasive infections, empiric therapy. Vancomycin is widely considered the empiric drug of choice. Clindamycin is an alternative if vanc isn't an option. Combination therapy with an aminoglycoside like gentamicin is often recommended, especially for critically ill patients. For synergy and broader coverage initially, directed therapy. Once susceptibility results are back, the MICs, remember, therapy is tailored. But duration is typically prolonged, usually 4 to 6 weeks for bacteremia, maybe 6 weeks or even longer for endocarditis, often requiring surgery too. And source control. This is absolutely non-negotiable. Any potential focus of infection, an infected central line, a prosthetic valve, an abscess, it simply must be removed or drained to achieve a cure. Antibiotics alone often won't cut it.
What about specific Dutch guidelines? You mentioned guidelines are scarce. Are there any national recommendations here in the Netherlands?
That's a good question. A review of available documentation shows no specific Dutch clinical treatment guidelines for invasive infections caused by B. thuringiensis, or the B. cereus group. Dutch and Belgian documents tend to focus more on public health, food safety, exposure assessment, not so much direct clinical management of invasive disease.
So what should clinicians here do?
Clinical practice in the Netherlands really should align with those international best practices and the extrapolated evidence we just discussed, using guidance from other European bodies like Public Health England and the UK, which strongly advocates for vancomycin as the primary treatment.
And what about our youngest, most vulnerable patients, neonates? Are there different considerations for them when facing a bean infection?
They're high risk, right? Extremely high risk. Neonates are a particularly vulnerable population for severe invasive B. cereus group infections, often linked to contaminated hospital environments, sadly. Empiric therapy. For a neonate with suspected or confirmed sepsis from this group, empiric therapy with vancomycin is the standard of care. It's generally effective and has a known safety profile in this age group. Dosage, frequency, and period. Now, specific antibiotic dosing in neonates is complex and must always be guided by local pediatric and neonatal formularies. You have to meticulously consider the baby's weight, gestational age, renal function. It's very individualized.
And CNS involvement.
CNS involvement, yes. A lumbar puncture is strongly recommended for all neonates with B. cereus group septicemia. There's a real risk of CNS dissemination. If meningitis is confirmed or highly suspected, antibiotic therapy needs to be extended to at least 3 weeks. And you might need to consider agents with good CNS penetration, like linezolid, or maybe cautiously, fluoroquinolones, always in consultation with pediatric ID specialists. Alternative agents, ciprofloxacin and linezolid might be effective, but they're often unlicensed for neonates. It requires a very careful risk-benefit assessment for each individual baby.
This brings us to a really crucial point for us in the lab. Antimicrobial susceptibility testing, AST. What are the key challenges and, critically, the EKCAST position on testing BT?
Okay, this is a big one. It's a significant gap in standardized interpretive criteria that, frankly, poses a direct risk to patient safety. The European Committee on Antimicrobial Susceptibility Testing, EUCAST, has not established clinical breakpoints for Bacillus thuringiensis or any other member of the B. cereus group. Let me repeat that. No clinical breakpoints.
That's surprising and worrying.
It is, and it's the single most important fact for any European clinical laboratory to know. Applying breakpoints from other organisms, like staph, is not recommended. It can lead to significant interpretive errors, potentially compromising patient care.
So if there are no clinical breakpoints, what should laboratories be reporting? What's the responsible approach here?
When no clinical breakpoints exist, EUCAST guidance is quite clear. You report the quantitative MIC value, the number, but without an SR interpretation. No susceptible, intermediate, or resistant call based on a breakpoint.
Okay, just the number.
And you must accompany the result with a mandatory comment. Explain that no official clinical breakpoints are available, and therefore the clinical efficacy of that drug at that MIC is uncertain. You can reference epidemiological cutoff values, ECOFFs, if they're available, but state clearly that an ECOFF is not a clinical breakpoint. It cannot predict therapeutic success.
So it forces a conversation.
Exactly. This approach necessitates a direct dialogue between the microbiologist and the clinical team to make informed therapeutic decisions based on the MIC value, the clinical picture, and the site of infection. Methodologically, disc diffusion. It shows high error rates and very poor agreement for B cereus group isolates. It's strongly discouraged for guiding therapy in serious infections.
So BMD then?
Broth microdilution, BMD, is a reference method and really should be used for all clinically significant isolates where you need an MIC. And remember, always remember, the fundamental interpretive rule of intrinsic resistance overrides any in vitro result. All B cereus group isolates, including BEAT, must be reported as resistant to all beta-lactam agents, penicillins, cephalosporins, carbapenems, regardless of the measured MIC. That's the crucial safety net.
Okay, we've covered identification, disease, treatment, testing. Finally, let's talk about infection prevention and control, IPC. The resilience of BEAT endospores sounds like a significant hurdle for healthcare settings.
Indeed it does. The core IPC challenge with the Bacillus species, including the peat, is that incredibly hardy endospore. These dormant structures are everywhere in the environment: soil, dust. They get introduced into hospitals easily, and once they're there, they're exceptionally resistant.
Resistant to what specifically?
To drying out, to heat, to alcohol, and this is a critical point of failure for many standard protocols. Alcohol-based hand rubs are not sporicidal, and they're resistant to many common disinfectants like quaternary ammonium compounds or quats.
That's a huge challenge for standard cleaning and disinfection. So what are the core IPC measures we need to adopt specifically for BEAT and other spore-forming bacillus?
Preventing healthcare-associated infections requires specific targeted measures that go beyond the usual: hand hygiene. Because alcohol is ineffective against spores, vigorous hand washing with soap and water is mandatory. After contact with infected patients or their environment, the mechanical action of washing is crucial to physically remove spores. Staff need explicit education on this.
Not just relying on the alcohol gel.
Absolutely not for spore formers. Environmental cleaning and disinfection. Routine cleaning is important, but for terminal cleaning of a room or during an outbreak investigation, you must use a sporicidal disinfectant.
Like what?
Effective agents include sodium hypochlorite, that's household bleach, typically at a concentration of at least 5,000 ppm free chlorine or hydrogen peroxide-based products like 7.5% H2O2 or vaporized hydrogen peroxide systems or parasitic acid-based formulations. You need something proven to kill spores. Patient equipment and devices. Reusable medical equipment needs meticulous cleaning first, then high-level disinfection or sterilization that is sporicidal. Think autoclaving, ethylene oxide, hydrogen peroxide gas plasma, and strict adherence to single-use policies for disposables is also essential.
And in terms of outbreak management, what should trigger an investigation, and what are the key control measures we need to deploy if we suspect one?
A single case of invasive B cereus group infection, especially in a high-risk unit like a neonatal intensive care unit, NICU, should immediately trigger an IPC investigation. Don't wait for multiple cases. Documented outbreak sources have included some surprising things: contaminated hospital linens, shared patient care equipment like thermometers, dust from nearby construction, even contaminated commercial products like non-sterile alcohol prep pads, which paradoxically can introduce spores instead of killing bacteria.
Wow. So what do we do during an outbreak?
Engineering controls are important. Maintaining HVAC filters, monitoring air pressure, promptly managing any water damage or leaks. During an active outbreak, cohorting patients might be necessary. Prompt removal of potentially contaminated indwelling devices, like central lines, even from patients who aren't showing symptoms yet, is often a key control measure. Surveillance cultures of patients or the environment might also be implemented. Look, the ubiquitous nature of borite means complete exclusion from the hospital environment is probably impossible. The focus for IPC has to be on minimizing its introduction and, most importantly, rigorously applying those sporicidal practices to prevent environmental reservoirs from becoming sources of patient infection.
What an illuminating exploration of Bacillus thuringiensis. It really highlights how an organism often seen just as beneficial, the pesticide, can pose serious, unique challenges in the clinical setting, especially for us medical microbiologists.
Indeed. Understanding its taxonomic quirks, its specific lab characteristics, especially that telltale perisporal crystal we mentioned, and the critical implications of its intrinsic beta-lactam resistance. Those are paramount. And remembering that our standard IPC measures, especially alcohol rubs, are often insufficient against its hardy spores. That's a really key practical takeaway.
This deep dive really underscores the importance of staying informed beyond standard guidelines, doesn't it? Understanding the nuances of microbial identification, resistance, and control.
It really does. And it raises an important ongoing question for all of us. How do we continue to adapt our knowledge and practices as new insights emerge about these complex organisms? Particularly when we're faced with this lack of definitive clinical guidelines for treatment or breakpoints for testing.
That's definitely something to mull over. We hope this deep dive has provided you with valuable insights and some actionable knowledge for your practice. We also invite you, our valued listeners, to provide feedback, share your own institutional experiences with managing Bacillus thuringiensis infections, or maybe contamination issues. You can do that in the comments section below. Until next time, keep digging for knowledge.